Hey everyone, welcome to the Peter Attia Drive. I'm your host, Peter Attia. The drive is a result of my hunger for optimizing performance, health, longevity, critical thinking, along with a few other obsessions along the way. I've spent the last several years working with some of the most successful, top-performing individuals in the world, and this podcast is my attempt to synthesize what I've learned along the way to help you live a higher quality, more fulfilling life. If you enjoy this podcast, you can find more information on today's episode and other topics at peterattiamd.com. Hi, everybody. Welcome to the fifth and final episode in the Week of Tom. This is the tour de force magnum opus course on lipidology. As we round this out, we talk about a number of things. We talk about Lp(a), inflammation, oxidized LDL, remnants, red blood cells and cholesterol, a biomarker called Lp(PLA2) that you've often probably heard about, another biomarker that you may have not heard of called ADMA, or asymmetric dimethylarginine, and SDMA, symmetric dimethylarginine. And then we kind of bring it back to what is one of the more tragic stories in Tom's life, which is a friend of his who unfortunately probably had FH, wasn't treated, and in many ways is sort of one of those patients that still kind of haunts Tom. And I think, we open this back in part one by talking about Tom's passion for two other things, firefighting and hockey, and how that passion is what really got him into, or basically allowed him to apply himself to this new field once he became obsessed. But also I think most physicians listening to this will have a similar story where there are certain patients or there are certain personal relationships where a loss of life stays with you for a very long period of time. And in many ways, it colors what you do over time. So I hope you enjoy this fifth and final installment of the lipidology advanced course. And next week, we'll be back to our regular scheduled programming where we're just doing, one podcast a week. So I hope you've enjoyed this, and I hope you find the show notes very helpful. I can't express how much work has gone into it. So without further delay, here is the final episode with Dr. Tom Dayspring. This will be a nice way to dovetail niacin, statins, and PCSK9 inhibitors. What lipoprotein have we not discussed today that would tie in a discussion of those three? Well, the astute listener realizes it's our good friend Lp(a). All right, why do I bring this up? So Lp(a), there are some people who still argue, hey, niacin makes sense because it lowers Lp(a). I'm going to save everybody on this podcast the brain damage of listening to us discuss that for the next 20 minutes and just bypass us to the more interesting question, which is, why don't statins lower Lp(a)?
Well, first of all, we need a better understanding of Lp(a) clearance. I'm personally of the belief that it is the LDL receptor. There is a plasminogen receptor that could clear some things, and there are macrophage receptors that, depending what else is going on, might internalize some Lp(a) and get rid of it that way. But it's the LDL receptor that clears it.
And also, let's pause for 1 second, because I realize even though we've already done an entire podcast on what is Lp(a), there's someone listening to this who hasn't heard it. You'll go back and listen to it, I'm sure. I'm going to do future podcasts where I interview the world's expert on the topic. But Tom, can you give in 2 minutes a description of what Lp(a) is?
Yeah, it's an LDL-like particle to which is adherent another apoprotein that shouldn't be there, and it's called apoprotein a. And you must pronounce it as a. That means small case rather than capital A. Lipoprotein capital A is apoprotein A1. That's the main apoprotein on an HDL particle. So if you tell me you got lipoprotein A, how do I know? Are you talking about you got HDL particles? Are you talking about a, which clearly identifies to me this potentially pathogenic, atherogenic LDL particle? So use the correct terminology with everything. And Peter's got that nice podcast on it. Our metrics of is that present in you or not is most labs will run an Lp(a) mass, which is the weight in a deciliter of plasma of the weight of all your LDL particles, everything that's in them, their triglycerides, their cholesterol ester, their phospholipids.
Meaning any lipoprotein that is carrying an apo lipo little
Yeah,
a.
and
You weigh
they're
the
all of the
entirety.
LDL density.
Yeah.
But you're weighing everything. You're not weighing apo little a. You're not measuring apo little a per se. Yes, that has a molecular weight, but the molecular weight of apoB is way higher than the molecular weight of apo little a. But you're measuring that too when you do an LP little a mass.
But the molecular weight of apoB is known because it's pretty much the same, whereas the molecular weight of apo little a is variable.
It's very variable, so that's another reason LP little a mass becomes a useless metric. And how do I know how many cholesterol, triglyceride, phospholipid, 10,000 other lipid moieties, other proteins are on that particle? I don't. And you have heterogeneous mixtures of particles that would have variable amounts of all of those constituents. So you can see there's going to be a weakness to measuring LP little a mass. Now, if it's really high, you've probably got too many LP little a particles. But since there's almost no more debate that the best metric for our LDL particles is low density lipoprotein particle number, there's no debate among those who truly understand that HDL particle number is your best available metric on quantifying the number. And that is a better metric than guessing that using HDL cholesterol or LDL cholesterol. Wouldn't you rather have a really nice finite count? If LP little a particles are dangerous, wouldn't you like to have a excellent, accurate concentration in them? Well, the only way you can do that nowadays, NMR provides those other HDL, and NMR cannot assay LP little a particles because NMR assays lipid content of particles, not protein content. So the only way I can give you an LP little a particle count is I have to separate all lipoproteins electrophoretically, and you'll see little bumps on the electrophoretic. VLDLs congregate here, LDLs congregate here, HDLs congregate there. And what's that little bump that is in this person, but it's not in that person? Oh, that's LP little a, because that's due to its surface charge where it migrates between the cathode and the anode and the gel that you separate them on. So if I then take that LP little a hump and I immunostain it with a antibody that binds to apoB,
You've just counted
I've
the
just
number of
counted
particles.
the number of particles that are in the LP little a distribution range. I theoretically should not call that an LP little a particle count. I should call it LP little a apoB. That's too much of a mouthful. It doesn't fit on a request form. So, of course, it's abbreviated LP little a -P. And to me, the downside of that is most people think it's an NMR measurement, which it absolutely is not. Basically, if you want that metric, and we've already announced I do work for True Health Diagnostics. I was working with a different lab before that, the lab that actually did the development of the LP little a apoB assay, which True Health absorbed and got proprietary rights to when he did it. So that's it. And believe it or not, there are people who are going to have discordance with LP little a mass, the weight of the whole kit and caboodle versus the number of LDL particle counts. In general, they correlate. But if you really want to know, and also I find it useful, at least those of you who are doing LDLP by NMR, you have your total LDL particle count. But what does that really include? It includes LDLs that don't have apo little a attached, and it includes LDLs that do have apo-A attached. Now, I throw a statin at you, and we started to talk about this. That statin is not going to budge your LP little a particle count at all. But the statin is going to dramatically lower your LDLs that don't have apo little a attached to it. So your LDL total particle count will go down a little bit, but part of that is your LP little a particle count. But this shows you are getting benefit when you give a statin to somebody with high LP little a.
You're going to have to go after residual risk. One of the best examples I saw of this was an unusual case where a guy had an LDL particle of 1,600-ish nanomole per liter, but he had an LP little a of 600 nanomole per liter. So even though it's an apples and oranges assay, directionally speaking, he's got 600 LP little a particles.
Or nanomoles of particles.
Nanomole per particles per liter, and 1,000 non-LP little a's. But when you give him that statin, you're really only targeting the 1,000.
Correct.
And so you'll be surprised at the lack of response you might see because you're not getting to target those 600.
No, but you'll still get your 30, 40. Yeah, but your total LDLP may still be higher than you wish it to be. And unfortunately, that is LP(a) particles. Then you can get into the theoretical discussion right now that if I could get rid of them, and basically the only way you can do that now is to maybe try niacin, which is a weak lowering of it 20%, or a PCSK9 inhibitor, which
seems like 30 to 50%.
Well, it would be an individual response, but it's in that range. Yeah. So if somebody could at least afford that drug, you can't get it covered by a third party because they don't have that indication to help you manage LP(a) patients. So, and that'll never come. Because could I ever prove to anybody that the PCSK9 inhibitor, because it's lowering LP(a), is reducing events? No, because they're going to turn around and say, all its event reduction is they're just getting rid of the remnants in the LDL particles. It has nothing to do with lowering. And how would you argue against that? We need a drug that only lowers apoC-III or LP(a) and does nothing to LDL particles. And that drug is under investigation right now. It's an ApoA synthesis inhibitor where your liver is going to stop making the vast quantities of ApoA that these people who genetically inherit that propensity.
This is courtesy ISIS.
Yeah.
Again, not to be confused with...
let's hold our breath. But so until then, are there people in the lipidology world who would say, Tom, until then, I'm going to continue to use niacin because lowering it has to work? I've been around too long saying lowers this and lowers that, and they don't work. So is niacin going to have the same toxicity that I think it has in other people in LP(a)? I don't know. But if you want to use that because you want to make a patient happy because you're improving some metric, be my guest. I would not. I know many lipidologists who would not. But there are ones, and look, Sam Semikis is probably the world authority on this. Sam uses niacin in his practice too. And I hope Peter has him on one day with a podcast. He would tell you, until we get our data on these other drugs, I'll try and lower it a little bit. If nothing else, he'll hash to it. I'm lowering ApoB a little bit too. And I don't know if it matters, but I'm raising whatever HDL metric. By the way, although fibrates are probably our best available drug now to raise HDL particle count, niacin, which blows away fibrates on raising HDL cholesterol, does nothing to HDL particle counts. I didn't mention that before. Because niacin makes the HDLs bigger.
I was just about to say, that could actually suggest it's worsening HDL function.
Yeah, you're right. So, but the niacin people will convince you that the big HDLs are protective. It's laughable, and the small HDLs are harmful, whereas the VA has
probably
real data showing it's the opposite, because the lipid-poor HDLs are the ones that can accept the most lipidation. So, you're never going to have definitive answers on any of that, but there's a lot of BS around.
So what's the best explanation for why a statin, which causes the liver to upregulate LDL receptors, does not lower LP(a), whereas a PCSK9 inhibitor, which also net results in more or a longer transiency of LDL receptors, seems to lower LP(a), even though that's not its primary objective.
And by the way, the data certainly tells us now that the main determinant of LP(a) mass or LP(a) particle concentration is ApoA production in the liver, not clearance. So until we can really inhibit production of ApoA, who knows what you're doing. But until then, let's in here. So how do LDL receptors clear these particles? Well, earlier I think I mentioned that LDL receptors looking for ApoE or a section of the ApoB that it can latch onto. There's a very specific area on the ApoB protein, on any of the ApoB particles, that because of surface charges, will bind to a specific part of the LDL receptor. That area on ApoB is called the LDL receptor binding domain. So if here comes an LDL particle and that domain is sticking right out, it'll stick right to any expressed LDL receptor. But what if there's something camouflaging that ApoA or that LDL receptor binding domain on ApoB, such as an interloper like apoprotein(a)? I could see where that would slow the clearance of an LP(a) particle because it's not going to bind as rapidly and as avidly to an LDL receptor. So most of these people have way too many LDL particles. We express whatever LDL particles we want. We try and express more with the statin. What are they going to grab first? the LDL particles that don't have apoA attached. And only then would they maybe then start even grabbing apoA particles or so. And do we all really have enough LDL receptors? Most of us, for a variety of reasons, do not. Other things retard the clearance too. You know, the apoC-III, I talked about other proteins that may be affecting clearance too. So there are just other factors at play. But right now, my guess is apoA is affecting totally efficacious binding to an Lp(a) particle. It's camouflaging the LDL receptor binding domain on apoB. And therefore, I better learn how to inhibit synthesis of it.
So Sam will obviously, hopefully we do get Sam on the show and we'll talk about this, but that this antisense oligonucleotide is basically going after the jugular issue, which is you inhibit the synthesis of apoA.
Yeah. And hopefully you do that. You know, the $1 million question has always been, why do we even have apoA to begin with? Did it ever serve some physiologic function, or is it important in any other aspect of human life? I guess we'll know it if we eliminate it, and there's some bad outcomes in that trial. And obviously, it absolutely will be looked at for every safety aspect anybody can ever think of right now as they're developing that drug. So right now, I don't think there's anything they're worried about. Sam would know more than I. But yeah, so if we can reduce that, and I don't know that we can shut it down completely, but he can drastically lower apoA and Lp(a) levels to, so, you know, everybody thinks either have it or you don't. There's a lot of people who have what's considered a physiologic or a non-adversarial concentration of Lp(a) in your system. So you don't have to make it go to zero, you know, where risk is concerned. In fact, the only people that are ever going to study this drug is in people like you just mentioned who have an Lp(a) mass or particle count up in that 600 range or something. They're not going to take people with borderline Lp(a)s and risk a clinical trial on them, just like the first statin trials were done in sort of FH type people or so.
Yeah, they're enrolling for phase 3 on that trial, aren't they? And is it secondary prevention or primary prevention?
I'm pretty sure it's secondary prevention.
Yeah.
And remember, there is some data in the apheresis world that if you pull out Lp(a) particles, there is they're not randomized blinded trials, because how would you do that with apheresis? That apheresis, for the non-physician, it's a dialysis where you're clearing your blood you take the blood out, you get rid of the LDL particles, and you give the residual blood back. You know, so, and for at least a week or two, they have less. But there's so many other things you're clearing, the regular LDLs, all sorts of rheological things, all sorts of other hyperosmotic proteins and things that who knows why apheresis really works, you know.
You know, I don't want to get us too far off our main theme of lipidology, but because they're both quite recent, let's just really, really briefly talk about two other interesting drug trials which look at a completely different component of the atherosclerotic mechanism pathway, which is it's the inflammatory pathway. So there was an agonist to IL-1, I believe, or maybe was it IL-1 or IL-6?
IL-1.
Yeah. And then there was also a trial that looked at using low dose of methotrexate.
Which is still ongoing. There was some preliminary words out of that this is looking good, but it's not been definitively
That's right.
released.
We won't know till the fall.
So, but there is the theory, reduce inflammation, and you will certainly help vascular health in capacities, maybe even have nothing to do with lipids or so. And so what's a great anti-inflammatory drug? Well, we've been using low-dose methotrexate in people with inflammatory disorders without a lot of toxicity for a long time, rheumatoid arthritis, et cetera, colchicine, very powerful anti-inflammatory. There's a big outcome trial undergoing with that. And I believe within the next year, we're going to have data on both of those trials. So, and the problem, this IL-1 inhibitor that Peter talks about, which has already published nice outcome data by inhibiting that, but it's a $30,000 drug. And there was downside to it because by seriously inhibiting the inflammatory system, there was a slight increase in cancers in some people. Your immunological system has uses in the body, so you better be careful how much you knock it out. Because, of course, and because of that downside, it will never get an indication for me and you to use it. Again, if you're some multi-billionaire, it's already been approved for certain conditions. You could use it.
Yeah, no, I think the bigger issue is really, as more and more of these drugs become available, hopefully it leads to a more and more personalized type of intervention where, look, if you got somebody who's walking around with a low C-reactive protein and a low fibrinogen, and their issue is elevated, LDLP, I don't see why you'd want to use an IL-1 agonist on that, or antagonist rather, on that patient.
Clearly not.
So yeah, it comes down to basically stratification of what's the driver. Again, this comes back to this idea of necessary but not sufficient, sufficient but not necessary, neither necessary nor sufficient. Inflammation is necessary but not sufficient. You do need an inflammatory response, but you can have an inflammatory response, and that by itself doesn't necessarily...
No, and a certain degree of inflammation is critical for you to get rid of certain pathologies or so. So you never want to stop the immunological system. Yeah, look, I believe, like Peter does, these are multifactorial diseases. We've spent a lot of time on apoB, LDL, but there are many other contributors that go into this. So I believe the better we can evaluate using biomarkers, the better we can identify many of the known existing pathologies. I'm sure there's many we don't fully understand yet. And then perhaps we can better individualize our therapeutic suggestions to these people and know what we have to do both nutritionally and pharmacologically, if that's necessary, to normalize whatever you're trying to normalize. So, yeah, it's a complex world out there, and you got to look at a lot of things.
So we've already discussed many biomarkers on the lipid front, and we've alluded to some of the biomarkers on the inflammatory side, C-reactive protein, fibrinogen. What are some other biomarkers that you find helpful as far as understanding even more diffusely endothelial dysfunction or other markers of oxidized states that can give us more of an indication that say, look, you might have two people with the same degree of dyslipidemia, but one has a greater burden of oxidation going on than the other, which would be presumably worse.
Presumably, yes. And the problem is going to be when you even start expounding on it, and I can throw some markers at you that you might want to measure that would tell you there's a pro-oxidative state going on. If your mission is to reduce atherothrombotic events, trials that show, oh yeah, if we improve this oxidative marker, that oxidative matter, people do better. They've never, ever been adjusted for apoB or LDL particle count. So even though you may show some nice data on them, if you really just normalize apoB and LDL particle counts, would it even matter? I mean, and don't tell me what common sense suggests. And look, I'm all for it. I use oxidative markers now mostly to try and convince people to do certain nutritional therapies rather than I got a wonderful drug for you to reduce inflammation, because right now I really don't. I know if I lower apoB, I reduce inflammation. So that's one, perhaps that's your first therapeutic initiative, lowering apoB by nutrition, or if the risk category is high enough, a drug, fine. But if could I measure something that tells me you have a pro-oxidative state? Yeah, and people do oxLDL levels in the blood, but they have no idea what they're measuring. Remember, an LDL particle, a truly oxidized LDL particle, which is the only type that a macrophage in your arterial wall can internalize, is oxidized in the wall of the artery. And what is oxidized? It's the phospholipids on their surface, or maybe some free sterols, be it a phytosterol, cholesterol. That oxidized surface lipids is what the scavenger receptor on a macrophage is going to pull that particle into it, and then you start accumulating cholesterol, and you have a steroid-laden foam cell. Bingo, you got the disease. So, but can I measure oxidized LDL in the plasma? No, LDLs don't get oxidized in the plasma. There's too many natural antioxidants in plasma where they don't occur. So what you can measure in plasma is what's called minimally oxidized. All right, there's a little bit of oxidation occurring, but it's not the phospholipids. It's some of the proteins on the apoB segment are being changed into aldehydes, which is what the monoclonal antibody picks up. It picks up aldehydes on apoB. But those aldehydes wouldn't have formed if there wasn't a little bit of a pro-oxidative state. So when you're measuring oxLDL, it's really apoB, a measure of LDL, apoB oxidation. But where is apoB? It's on remnants and on LDL particles. So just understand what you're measuring. But of all the LDL particles that are floating around, if you get an abnormal you're looking at maybe 5% of the total particles. Now, you might make the case that when they do enter the artery wall, forced in by the particle number, they're going to be even further enhanced. So maybe they got a leg up on an unoxidized LDL particle. Okay. So I would use that as a measure of a pro-oxidative state, and I'd try and give you whatever nutritional things I believe might fight oxidative states or so. I'm not knowing that I got a drug, including a supplement, that ensures me it can do that or not, other than I would, if I had to reach for a supplement, it might be an omega-3 fatty acid, but it's another story. So that's what you're measuring with the oxLDL. I think there are better markers of pro-oxidative states. And look, a lot of oxidation is the granulocytes oxidizing a variety of tissues. So myeloperoxidase is a granulose-secreted pro-oxidative enzyme. We can measure that easily in the blood. That would be a signal of a pro-oxidative state. And I think the best of all is as fatty acids get oxidized, derivatives form. They're shorter chain fatty acids, and they're put in a big group called isoprostanes. And you can easily assay something called F2 isoprostanes, which have clearly been linked to pro-oxidative states. Now, it's a urine test. It's not a serum test, but it's a small urine sample. It's not 24-hour urine. So I like to do F2 isoprostanes. So whatever you like to do, be it oxLDL, be it myeloperoxidase, or be it the F2 isoprostanes, I can use that as useful information and maybe even convince you that nutrition is important here, and that is the best way to try and change them around or so. So those are some of the pro-oxidative markers. What I would not use as a pro-oxidative biomarker that way too many people is lipoprotein phospholipase A2. First of all, the data is pretty poor. Most people don't even know. The only thing the FDA has given that approval for is as a screening test in primary prevention, never ever for anybody on a lipid modulating drug or on who has known coronary atherosclerosis, for the simple reason that Mendelian randomization has shown your level of Lp(PLA2) activity or mass has nothing to do with outcomes, sort of like the HDL cholesterol story. And even more important, there have been 2 mega trials where pharmaceutical industry has developed Lp(PLA2) inhibitors, gave them to patients, shut down Lp(PLA2) activity, drastically reduced Lp(PLA2) mass, levels plummeted, no outcome reduction whatsoever.
And Lp(PLA2) is an enzyme made by the endothelium.
No, it's made by macrophages. It's a macrophage. It's just like myeloperoxidase is made by granulocytes.
But the MPO data are equally unimpressive, aren't they?
Well, if you want to come down to, it depends what you're using it for.
I stopped using MPO like 2 years ago.
So look,
and I agree with you. I think I
don't do it myself. I do the oxLDL. I do it. It's easy to do it.
Yeah.
It's there, and it's more linked to the process that I can see. MPO, I don't know. You could have some other vasculitis going on or something.
Yeah, my take on MPO is I found it to be unhelpful, and I agree with you that Lp(PLA2). It's one of those things you only really need to check once. Where I find it actually somewhat helpful, and again, I could be just deluding myself, is you take the patient with an elevated Lp(a) who has an elevated Lp(PLA2), and not only do you have a tongue twister, but you also have a patient that might be a little higher risk.
And I wondered if I whispered that into Peter's ear over the years.
You may have. You
but
may
anyway,
have.
I would tend to agree with him. So here's why. Lp(PLA2), which is an enzyme that does oxidize those phospholipids on an LDL when, after that LDL enters the arterial wall, is exposed to reactive oxygen species, and some of the fatty acids on the phospholipids start oxidizing. Then Lp(PLA2) activity kicks in, and it deesterifies the fatty acids from the phospholipids. So now you got oxidized phospholipids floating around and a byproduct called lysolecithin, which is a pretty pro-atherogenic molecule. So Lp(PLA2) is involved with the production of them in the arterial wall. Well, that's not where we're measuring Lp(PLA2). But, hey, if it's on the particle when it goes in the artery wall, whose artery's wall in today's nutritional environment doesn't have reactive oxygen species sitting there waiting to attack something? So, boy, what particles entering your artery wall are going to have the most Lp(PLA2) mass on it, when that's where you're going to turn on Lp(PLA2) activity. And by the way, the new assays available everywhere, they no longer measure the mass, they measure the activity. We were the last company who finally ran out of reagents, so we're going to activity now. So across the board, The studies show either LP, PLA2 mass or activity are identical in predicting risk if that's what you're using it for. But believe it or not, the activity assay is so much easier technically to do, less costly. That's what all labs are doing nowadays, the activity. So back to LP-PLA2 jumps on LDL. It's a little bit on HDL particles, but most of it is on HDL particles. But HDLs are a heterogeneous species of normal size, big, and small LDL particles. You'll find way, way more copies of LP-PLA2 on the smaller LDL. Perhaps one of the reasons, if people believe small LDLs are more atherogenic than larger sizes, I don't know, once they're in the artery wall, which is driven pretty much by particle number, but everybody with small LDLs has probably got too many LDL particles. So they're very prone to oxidation when they do go in the artery wall. Not that a big LDL can't be oxidized, it could be, but it would have less LP-PLA2 on it. But if I really wanted to know the number of copies of that enzyme, LP-PLA2, where would I find it in the blood? On LP-little-a particles. That's where it's all carried. So I think Peter hinted on it in his LP-PLA2 podcast, that there's another attribute to LP-little-a other than quantifying it or weighing its mass, and that's, we do know there are people with high LP-little-a who do not get out. They may be a minority, but they're out there. Just like not everybody with FH is going to have a heart attack. Some go through life without it. Most get some events, but many do not. And clearly there are people without LP-little-a who get heart attacks for other reasons. So if you came to me with a very high LP-little-a whatever, do I just say life's over, get your will in order, I got no drugs that can really help me with this, and you can't afford a PCSK9 inhibitor, so I can't even experiment there? Maybe the ones who are at most risk, part of the functionality, part of the atherogenic potential of an LP-little-a particle is one of the functions, perhaps, of apo-little-a is it's a garbage truck that scavenges oxidized lipid moieties, phospholipids, sterols, and it brings them back to wherever. Maybe it's macrophages that clear them and detoxify them. So it's some sort of a little rescue truck, garbage truck of clearing it. But what happens if you got way too many LP-little-a particles, too many LDL particles, so some of them are going in your artery wall? Now you have an LP-little-a particle that's not only got a potentially pro-atherogenic apo-little-a protein on it, but that thrombotic, if it is a prothrombotic protein, there's plenty of evidence suggesting it is. By the way, with arterial, not so much venous thrombosis. Many people have dismissed LP-little-a as being linked to venous thrombosis. I don't know what the story is, but if you talk to Sam on that, he will tell you.
Oh, that's interesting. So I stand corrected because I think I was probably quoting some old data when I said that the VTE hazard ratio is about 2.
Yeah, but it's just weak sort of data. So when you talk to Sam, talk to more about that. So maybe it is, maybe it isn't.
Athero, but aortic stenosis.
Athero first. Yeah, and aortic stenosis. Yeah. And part of the pathology, maybe it's just trafficking these oxidized lipid moieties into your particle, which is going to just stimulate this whole inflammatory mess that's going on in your arterial wall. And maybe it's a prothrombotic mess if that's a prothrombotic protein too, the double whammy going in with it. So, and I think Peter covered it nicely in his discussion there. There are these segments on apo-little-a called kringles, and there are a couple of kringles that have a large essence of lysine amino acid that is like flypaper for oxidized lipid moieties. So they really bind to these lysine-rich segments on apo-little-a. So if we could start measuring oxidized phenomenon and tying it into
now, Sam has written about this in his papers.
Well, yeah, and Sam has published the data showing that's who you worry about with LP-little-a. If they have oxidized ApoB phospholipids elevated, and Sam's got an assay for that. It's not available widely.
Yeah, that's a shame.
And Sam will tell you that, yes, we're measuring it on ApoB particles, but the ApoB particles that carry the vast majority of these oxidized are LP-little-a particles. Yeah, it could be a remnant or two or the oddball LDL particle, but mostly it's your LP. So that is, again, a double whammy. So I think maybe in the future we're going to be screening with whatever you want, LP-little-a mass, LP-little-a particle count. I hopefully never LP-little-a cholesterol. It's like all cholesterol metrics should be. And the only way you can assay it is a poor assay nowadays anyway. is, all right, you do have excess LP little a, whatever in your system. Let me now do this follow-up test. And if that's also up, let's discuss everything we can possibly do until that miracle drug, hopefully miracle drug comes along and we got a cure for you. And maybe it'll be covered in those people because the data will be strong. Because like all these new antisense drugs, it's going to come with a price.
Yeah. Before we leave biomarkers, anything you want to say about asymmetric or symmetric dimethylarginine?
It's a very interesting biomarker also, where we talked about endothelial function, perhaps even some of the antioxidant function is this miracle molecule every endothelial cell makes, extremely transiently makes it and it's gone. It's nitric oxide. Nitric oxide is a powerful regulatory molecule that regulates vascular reactivity, but oxidation, the thrombotic potential of an endothelial surface initiating a thrombus is highly dependent on nitric oxide. So when endothelial cells make nitric oxide, which is by far the most important molecule they make, where does that come from? Arginine, the amino acid arginine. So arginine gets converted into nitric oxide. So last thing I want is things that are going to screw up arginine pools, which is needed to make nitric oxide. And two proteolytic molecules, we know, byproducts of basically catabolism of nuclear proteins is something, one is called symmetric and the other one is called asymmetric dimethylarginine. They're isoforms of one another. So if they're mirror images of one another, but they're actually different molecules. One directly and the other indirectly inhibits the synthesis of arginine. So if I measured ADMA or...
The synthesis of nitric oxide, you mean?
Right, yes.
Yeah.
Well, ultimately synthesis of arginine, which results in the synthesis of nitric oxide.
Oh, see, I always thought that SDMA inhibited the synthesis of arginine, but ADMA inhibited NOS directly.
I should correct what I said. One inhibits the synthesis and the other enhances the catabolism of arginine. So at the end of the day, you're going to have less
nitric oxide.
And less nitric oxide. And they're both markers that we can easily measure. So, and if we measure them and they're high, you might presume, hey, maybe that's a blood test that actually tells us something about endothelial function. I don't think we have another that has anything close to at least that hypothesis.
I think the nice thing about the ADMA, SDMA is it creates a biologically plausible mechanistic explanation for why we see an association between high homocysteine and greater disease, because homocysteine really inhibits the clearance of ADMA and SDMA, and that's very clear when you correct it.
Which would therefore screw up nitric oxide production.
Correct.
Yeah, so it brings even oxidation into the process would disturb that process too. So if pro-oxidative state, if hyperhomocystinemia is adversarial to the vasculature, that's one of the pathways in which it is. So what can we do? Well, we can measure it. So who would I measure ADA? So if you came to me and you're on your third bypass or fourth stent, I think it's a pretty safe assumption you have screwed up endothelial function. So I don't.
And of course, the bigger question is, what do we do about it? So one thing is, look, if they have impaired renal function, hypertension, those are things that you go after. The homocysteine, especially in the MTHFR mutation patients, you give methylated B vitamins, which is still controversial in sort of mainstream circles. Because even though it lowers homocysteine, some will point to that and say, well, you have no outcome data on lowering homocysteine.
No, but you have plenty of data that in people with hyperhomocystinuria, lowering homocysteine drastically improves their vascular outcomes. So it's plausible at a certain threshold of homocysteine that there's probably makes sense. And if you really understood the pathway of catabolism of methionine, you'd understand this better. And remember, in that pathway is going to be the production of a lot of neurotransmitters too. So there's a lot going on if you have the genotype that's going to screw up methylation or the enzymes involved with that. That's another story. But perhaps the best use of ADMA, SDMA is in the primary prevention setting where, all right, you got a little borderline ApoB, LDLP. You're not going to do lifestyle. And do I really want to throw you on a drug or something? if those markers are up, that tells me whatever is going on in you, you have endothelial dysfunction. So you're down the road towards vascular pathology. So it might be a marker to kick you in the rear to at least be super aggressive nutritionally. Even, hey, I'm sorry, of whatever nutrition we tried, you still have abnormal endothelial function. It's time for us to... Here, I would rather improve these markers that will take a drug to get them to where I want them to be. So maybe we can use it in that setting. If you also want to tell it to me, Tom, you said, hey, if you've been through a bypass or a stent or something, why do it? But what if I have such a patient who I've maxed out on a statin ezetimibe, I've done everything else to homocysteine or insulin resistance, and they're still up? Should I lower ApoB even further with a PCS? Because, I mean, you use it for what you want to use it for, but as a marker of endothelial function. And very interesting, of the two, you get both. And you also get an arginine level in the same assay. And SDMA is cleared by your kidneys. ADMA is catabolized in the cells. So kidney function has nothing to do with ADMA. But SDMA, one of the things that'll elevate it is renal disease. And when SDMA goes into serum, it can backflux right into the cell and decrease nitric oxide production, probably part of the pathology in people with renal failure.
I was just about to say, the thing I like about the ADMA, SDMA stuff, even though we don't have all this great long-term data, is it's one biochemical pathway that provides very clear evidence for two observations that are undeniable: elevated homocysteine and renal function.
Yeah, the SDMA, not the ADMA.
But you get both out of the...
You get both. It's not like you're ordering them selectively. You get them all three of those markers.
But they're addressing both sides of that equation.
Yeah, so it's a very interesting little biomarker there. And hey, who doesn't want another renal function test? Most are using creatinine. That's kind of a poor. You should be using cystatin C.
For sure.
And SDMA will be further enforcement that... And mild degrees of renal impairment go ignored in this country. And if you're even in the early, even stage 2, but certainly by stage 3, which could be a clearance of 70 or something or 60, people that gets ignored, even though EGFR is being reported to people. And that's a major league risk factor that you ought to say, you're a vasculopath, you're at risk for vascular disease. I got to see what I can do to you therapeutically to lower your risk. But it gets ignored until a creatinine clearance is 32 and you need dialysis or something, you know?
Yeah, this is one of those things I do talk about with a subset of my patients, especially these young patients in their 40s, 50s who buy cystatin C and creatinine and maybe even a touch of microalbumin. You know, their GFR is 80. And I say, if you're 40 and your GFR is 80, there's a problem if you want to live to be 100, because I don't know where you're going to be at 80.
Yeah. And look, if you got albumin in your urine, you got a serious vasculopathy someplace. Maybe it's just your glomerulus and you got a kidney disease, but you probably have some other vascular disease too. So albumin is just asking something else. If something as big as a protein's escaping in your urine, you got some membrane problems that are lining some either a glomerulus or a blood vessel someplace. The other ones are, and it's interesting because Peter in his little world sees a lot of these people who are well-trained athletes, take care of themselves, and some of them, and Peter probably encouraged them to build up muscle mass, and which can influence creatinine and makes it far less usable as a marker of renal. Cystatin C has not influenced by muscle mass, so that's where it picks up some of the imperfections, the weaknesses.
We check both.
Yeah, and if you do both, which is, by the way, if you read the kidney guidelines, they've actually developed a nice equation where your clearance, and we provide this at THD, and I don't know any other lab that does. We give you a clearance based on creatinine, we give you a clearance based on cystatin C, and we give you the best clearance of all based on both of those parameters.
And then you segregate by African-American, non-African-American.
Because creatinine is very different in African-American. So there's a lot more to those EGFRs, if you haven't studied it lately, that might be going on. And if I'm going to do a biomarker, if there's no other restrictions, and look, that might be a third-party payer or somebody else, I'd rather do the most informative biomarker, and that would be the dual EGFR and maybe some ADMA or SDMA thrown in as a renal marker anyway. And that SDMA ADM marker, I really wish we could take you through. Just be 5 or 6 PowerPoint slides, and you'd see these pathways. And maybe I'll tweet them in the next week or so, individual
Yeah, or we can
slides.
just attach them here easily to the show notes.
And you'll really, it's not a highly it's stare you right in the face how they work and what they're doing.
Now, we touched on something earlier that I know you wanted to come back to, and we were deliberately short on it, but I'm trying to kind of think about how to land this plane here. You want to go back and talk a little bit about what a remnant is? It gets so much confusion about remnants. They're talked about like they're one homogeneous entity.
Yeah, like there is First of all, every known lipoprotein class, including Lp(a), has remnants. All a remnant is a smaller part of its sister particle that's in the same density class. So VLDLs come in multiple sizes. You got the big ones, which are only found in people with triglyceride abnormalities. If you don't have a triglyceride disturbance, you will never have a big VLDL particle. You'll have medium size and very often just small LDLs. But if you look at your LDLs, there's a normal size particle, there's big LDLs, and there's small LDLs. Everybody has a heterogeneous mixture. There's no human on the planet who has 100% exact diameter of every single subfraction of a HDLs vary widely in their density and their size and their diameters. Even IDLs, there's a certain diameter range or density range where here's the upper limits of what you would call an IDL, and here's the lower limits of when do you not call it an IDL, it's an LDL. Or when does a small VLDL, I can't call it a small VLDL anymore, it's called an IDL. But wait a minute, wouldn't an IDL be a remnant of a VLDL? If that LDL has a VLDL or IDL origin, isn't an LDL a remnant of a IDL or maybe a VLDL? Chylomicrons, as they lose their triglycerides and phospholipids, they become smaller chylomicrons. Chylomicron remnants, which are, for the most part, if you're lucky, cleared by ApoE receptors somewhere, but there's chylomicron remnants floating around. So what is the only particle I really care about? An ApoB particle that can wind up in your artery wall, get oxidized and deliver the sterols, and you oxidize them, and then you got plaque. So what forces them in? It's particle number. Now, I've also explained, all right, if that's it's ApoB particle number, LDLP is your first biomarker. ApoB is simply an LDLP biomarker. You're not counting VLDL particles because there are so few of them. You're certainly not counting chylomicrons because there are even fewer of those. So ApoB is identifying to you way too many LDL particles in this person. But there is absolutely no doubt, even though you have way, way more LDL particles than you do have VLDL particles, because VLDL particles are 2 or 3 times bigger than an LDL, the volume of a spear is the third power of the radius, they do carry more cholesterol molecules per particle than an individual LDL particle. But of course, you have so many more LDL particles than even VLDL or whatever you think a VLDL remnant is. Most of the cholesterol getting into your artery wall is still LDL delivered, but I am not denying that VLDL particles cannot deliver cholesterol and get oxidized when they are into the artery wall. They can. So what has been the classic definition of a VLDL remnant? Well, they've always said, well, do that conversion factor we talked, divide triglycerides by 5. If that's high, the VLDL cholesterol is high. They have too many VLDL remnants. And no doubt, if we really had a VLDL remnant test, that's true. But it would not be true in other people. So it's a poor man's perhaps estimate of remnants.
And trying to clarify what you're saying, right? Because I think it's a bit confusing to the listener. You can estimate VLDL cholesterol in 2 ways. You can take triglyceride and divide by 5, and the higher the triglyceride level, the less accurate that becomes. You can take non-HDL cholesterol and subtract LDL cholesterol from it, and you'll get an estimate as well. Those two don't often agree, by the way, but nevertheless, you have 2 estimates. But that still doesn't answer the question.
Right.
Which of those go on to become pathologic remnants?
Correct. So all you're identifying there is you have increased cholesterol content in VLDL particles. And because the ones we fear the most are remnants, we're just presuming, oh boy, there's got to be remnants. There's got to be remnants here. So whatever therapy you're thinking of doing in this person, you better get rid of, apply some remnant therapy too. Well, all of the FDA-approved drugs that lower ApoB, whether it's statin, ezetimibe, fibrates, which are approved to lower LDL cholesterol, even though they don't really do it that much, PCSK9, clear all ApoB particles, including remnants. So your ultimate solution, now your first solution might be nutritional if it's not somebody coming off their bypass who is in the nightmare risk range classification where I think you need a lifestyle and pharmacology day one. You just don't have too many decades have passed. You shouldn't be putzing around other than a real short time. You know, the nutritional therapy, if I think you have remnants, is going to be, I'm going to address insulin resistance in you. And so I'm being an advocate of some degree of carbohydrate restriction and certainly a clear advocate for a lot of reasons, apart from even ApoB, would be the fasting as part of your diet and too. So great. If you want to use your VLDL cholesterol and it makes you think there's remnants and you're going to suggest the therapy, I would tell you if you're total drug guided, I have a drug, then that's the person you give a fibrate to with your statin, because that's what the fibrates really do well. But I can show you data ezetimibe, the statin clears as many remnants as that combination too. But I think fibrates bring other things to the table that perhaps Zetia doesn't in people with triglyceride-rich lipoproteins. And one of them is going to be, so it's how I really think we're going to have to identify remnants. So just even tell, there's a laboratory that produces, they separate VLDL particles by ultracentrifugation. The NMR people used to report total VLDLP, but they would also give you large VLDLP plus medium VLDLP plus small VLDLP. And we used to say, well, the small VLDLP, those are the remnants. That's the mark. Well, they don't do that anymore. They only give you the large VLDL-P anymore. Those NMR generating labs that have the proprietary ability to release VLDL particle data, and that's not all NMR labs.
Is that LipoScience or LabCorp
now? There is no more LipoScience. It's LabCorp. So you will get a large VLDLP. And it has one reason and one reason alone. It's a lipoprotein marker of an insulin-resistant state. That's it. It's not a goal of therapy. It's nothing. There's no data that what you do to VLDLP affects outcomes, although I'd make the case that maybe it does. But it's just an IR marker. I can measure small dense LDL cholesterol, HDL2 cholesterol, just small LDL particle concentration. I can do an insulin level and 10 other tests that tell me you're insulin resistant. So I don't need any VLDLP marker.
I mean, they were using that as part of their IR algorithm score.
If you don't have that, and they factor in VLDL size there, because the big triglyceride-rich VLDLs are a marker of insulin resistance. But if you're measuring insulin levels and other insulin biomarkers, they're no better than that. I don't know that they're any better than just measuring small LDL particle concentration, at least in a drug-naive person. So fine, it is a marker of insulin resistance. But this other lab, the Centrifusion, they tell you, look at our smaller VLDL cholesterol. Those are remnants. How do they know? All they know is that person has extra cholesterol in their small VLDL particles. But VLDLs, depending on two things, its ApoE content and its apoC-III content, is going to be virtually instantly cleared. So if your small VLDL particles are being rapidly cleared by your ApoE, ApoB100 receptors, what do I care? So in other words,
if you took a bunch of small VLDLs, and some of them had lots of ApoE and very little apoC-III, those aren't remnants. They're not pathological. They're not sticking around.
They're small VLDLs. That's what they are. But are they disease-causing VLDLs?
Right.
Look, some of them probably get in, but very few compared to LDL particle number. So, and also remember...
And very few relative to a small VLDL that would be high in apoC-III and or low in ApoE.
Never going to be cleared. apoC, the main there's a lot of downsides to apoC-III. It's a pro-inflammatory protein. It really enhances VLDL production in the liver, but it just retards their clearance, and it probably interferes with the hydrolysis induced by lipoprotein lipase. So you're increasing the half-lives of all triglyceride-rich lipoproteins, which lets CETP exchange occur longer. And what is CETP exchange doing? Bringing more cholesterol into your VLDL particles, enhancing cholesterol enrichment. Those VLDLs are losing triglycerides but gathering cholesterol. So these are all the pathologies that might go on if you have delayed clearance, and C3 is going to delay the clearance through multiple reasons of these particles. So I want a blood test that tells me the apoC-III content of your ApoBs. LDLs that have apoC-III cannot be cleared. In one of the early Pravachol trials, the pravastatin trials, the CARE trial, they went back and who with high triglycerides gets coronary events? And this is Frank Sachs up in Boston. And they've been able to measure this in sophisticated lipid labs for a long while. If your LDL had apoC-III on it, pravastatin was a useless drug. Didn't reduce any events. So what?
It still reduced ApoB, but it didn't reduce events?
Yeah, it would clear some LDL particles. It didn't reduce events in people who had high triglycerides. So the apoC-III, it's not measuring triglycerides that matters, it's apoC-III. And a lot of people with apoC-III might only have a triglyceride of 110, 120.
Wait, which trial was this?
A CARE trial, cholesterol and acute. It was the second secondary prevention trial. Simvastatin 4S was first, and that was Swedes with super high LDL cholesterol and heart attacks. This was done in Boston at the Brigham, where they enrolled people with unremarkable, at the time, LDL cholesterol 130 or lower who had heart attacks, and they gave them pravastatin and got this pretty much the same event reduction as they got in the 4S trial.
But the ones that had high trigs didn't have the reduction?
Yeah, so they went back in a post hoc analysis, and those who had high trigs, Pravachol lowers triglycerides. Does it matter? That's one of the benefits of pravastatin. Well, the only people that got event reduction where pravastatin lowered triglycerides was those who had apoC-III enriched LDL particles. So apoC-III becomes your biomarker.
Is Sam's company also working on?
I believe, in production is an antisense molecule that's going to stop apoC-III production. Because, Peter, I believe you also know the Mendelian thing there is gain of function, loss of function of apoC-III is tied into longevity also.
Actually, I think this is, of all the major longevity genes, I do not believe, and I could stand corrected, but I'm in the process of writing for the book now. I don't think there is a longevity gene on cardiovascular disease that is a stronger predictor of longevity than hypofunctioning apoC-III.
Yeah. So I'm very interested. I tweeted about it, I'm sure within the last week, a very interesting trial that if you're going to use an omega-3 fatty acid, when they compared DHA versus EPA, DHA is the one that lowers apoC-III, not EPA. So all of these mega advocates of EPA, I don't know if apoC-III is involved there. Maybe you better.
Well, I got to be honest with you. I used to see myself as more of an EPA guy. And then my focus on DHA, I think, became more something I saw in the mild cognitive impairment literature, which was the importance of DHA in the brain. But I didn't realize this DHA-C3 relationship. So
there, look, from my mind, for years, it's been omega-3s are not lipid drugs. Ignore them. Don't use them to putz around with lipids because you don't know what they're doing. That's changed now. Clearly, high-dose prescription strength EPA can be a helpful adjunctive therapy to lower ApoB. And whether it's with Remnants or whatever it's doing, it's lowering ApoB an additional 8-10%. Great, and it's a pretty innocuous therapy. I always worried about those who might not be able to convert EPA to what I believe is the necessary DHA also, especially in the brain. So, but I can measure that in the plasma.
You know, I've been thinking about, I'd like to have Bill Harris on the show as well to have a discussion about this because
you really should.
At least at the time of our recording today, I would say that the mainstream view of EPA and DHA is that they're useless, right? That's the press for today.
That's the mainstream view.
Yeah, the mainstream view is abandon all of these dumb sort of supplements. But as is often the case, when the mainstream says something in a declarative fashion, they're usually wrong.
Especially on a nutrient, where they're looking about what you're eating, not taking pharmacologically, where you might have a real serious trial.
They're confused by supplements versus pharmacologic grades.
Most of the trials that they would quote as omegas being useless, they're using some not a pharmacologic dose of these things. And they're not measuring even who's deficient in omegas. And if you're not deficient in it, why would I even give you an omega-3 product or so? And measuring fatty acids in the blood, I would encourage you to measure red blood cell omega-3s that are like a glycohemoglobin, have a 30 or a 60, 90-day half-life, rather than a plasma-free fatty acid or omega, which might be what did you eat for lunch. So there's other intricacies involved. But nonetheless, just all things to think about there. So in this DHA with apoC-III is kind of interesting. Now, look, some people, if you're just going to drown them in EPA, do convert it to DHA, so it's not an issue. But we can measure that, and I think we'd know. Oh, boy, and you got to have some of that either eaten or supplemented,
Are you pushing patients towards closer to 10% red blood cell EPA DHA level these days, or are you?
Well, I don't see patients anymore, but if you want to ask me, yeah, that would probably, there's no, Bill Harris would tell you, why not make it 14%? But we don't have any data that would show, hey, that's harmful, but we don't have any data showing you can even make it that high or so.
Not even epistaxis and things like that? Easy bruising?
So far, anything with it, they've never turned anything from an omega-3 into coagulation disturbance. I mean, it's old data that, hey, some Eskimos had a bloody nose one day on it, and what else? They're eating EPA and DHA all day long. So I don't know. That would have to be tested in a clinical trial.
Tom, I just have to take issue with that as a Canadian. I think we refer to them as Inuit now.
Oh, I'm sorry. Yeah. Eskimos is an old New Jersey term.
Super culturally insensitive.
Oh my God.
I'm not gonna stand for it.
I'm an old Jersey, right? So you're right. I insulted a lot.
But I really do need to get Bill on the show, because Bill's one of these guys who is, and again, many of the listeners won't know who Bill Harris is, but I hope to change that. This is a guy who is as devoted to understanding the entire body of literature and science of omega fatty acids in general,
especially omega-3s and 6s.
As you are to lipids.
Oh, way more than me. And he was a colleague I worked for 3 or 4 years with. I love the guy. We'd lunch all the time. We go out. So he has taught me so much. And he is the guy who developed the red blood cell phospholipid fatty acid assay. So he knows what he's talking about. And it gets so intriguing, because even for those of you listening who may be taking an omega-3 or prescribing an omega-3, be it a supplement or be it a prescription product, what vehicle is that? Are you prescribing a free omega-3 fatty acid? You're not. There is an FDA-approved product that the company who developed it has not brought to market yet. So you're either prescribing a esterified omega-3 fatty acids, or it's esterified to something else, or it's esterified to glycerol as a phospholipid, or it's esterified to a glycerol backbone where it has 3 fatty acids, but only one of them is an omega-3? Or are you buying a super enriched that all 3 fatty acids are esterified to an omega-3? You would need very small. So what is the vehicle in your favorite supplement that's carrying the all of that comes into the pharmacodynamics.
I just realized, I don't actually know because there's only 2 supplements over the counter that I fancy. One is Carlson's, the other is Nordic Naturals, just based on some of the toxicology stuff. But I don't know the answer to the question you just asked.
You can't get that information unless your brother works there and is privileged to that. The companies won't even tell you what they esterify. And look, you hear about these krill products. They're phospholipids. And that means there's 1 fatty acid on them. So you got to prescribe a lot of krill pills to achieve a certain omega-3 level. Way less than you would if you had a diesterified or luckily enough a triesterified triglyceride vehicle.
I'm always happiest when I see my patients just eat enough salmon.
Yeah. So listen, at the end of the day, that's all cool. It's good to know. But at the end of the day, whatever way they're somehow getting omega-3s into your body, if you're normalizing the omega-3 index, does that even matter, all that other stuff? So it all depends. But I would implore you to follow up, please.
Yeah, I want to dig into this a little bit, because it never occurred. I got to be honest with you. I'm ashamed to admit this, because I hate being the guy who makes dumb assumptions and is too lazy to follow up. I've always assumed that the high-quality EPA DHA supplements were triacylglycerides.
Almost none of them are. They're mostly monoglycerols, but it's glycerol with 2 other fatty acids plus 1 omega-3 fatty acid.
Yeah.
And what are those other fatty acids? If nothing else, they're calories that you may or may not need, or they're a harmful fatty acid even or so. Who knows?
Well, we'll get to the bottom of this. And there's no pharma-grade DHA, to your knowledge?
No.
Okay.
But here's what there is, and it's already FDA approved, but the company that developed the product is not brought to market. But I think they're going to, because this November at the American Heart Association, you are going to get the people at risk who are on statins, and half of them are getting statins plus EPA, and the other half are getting statins plus EPA placebo, no EPA. And we're going to get outcome data.
What dose of EPA?
It's high. It's 3-4 grams. It's the prescription strength. So are we going to see cardiovascular? All indications, and we're all optimists, are it's going to be a positive trial. They haven't stopped it, at least for futility. They certainly haven't stopped it for toxicity. The trial is over. They're generating the data now. Wouldn't they have just stopped the fertility and gone away if it didn't work? So we're all expecting that it's EPA added to the statin is going to work. Why wouldn't it? It's an EPA. It probably has other physiologic attributes that can reduce certain inflammatory markers. Hey, and it's a nice ApoB additional lowerer. Now, the company that has developed free omega-3 fatty acids, so they're not esterified to anything. That means they're very bioavailable. You just swallow it. Remember, anytime you swallow an esterified omega-3 fatty acid, you have to secrete pancreatic enzymes that deesterify it because it can't be absorbed without that. Even phospholipids, krill oil, has to be deesterified by a specific lipase coming out of your pancreas.
Free fatty acid is
free fatty acid is going to come right in, but we don't eat you might in certain foods, but even most of the food you eat, it's triglycerides that are delivering.
Don't free fatty acids taste horrible?
I don't know that I've ever. Maybe that's why they're not there. Or I think most of the time they're stored as energy and as not free fatty acids, and so they got to be deesterified. But anyway, this company that has them, why didn't they? Because this is a company that manufactures a statin, and what they were hoping was they thought the people making EPA, the FDA, was going to give them an indication based solely on additional LDL cholesterol lowering or ApoB lowering that will let you come on the market pending your outcome data because non-HDL is better, whatever. And the FDA didn't. FDA even told them they couldn't do it. The company, which is Amarin, actually took the FDA to court, and the Supreme Court says, no, even though you don't have an approval for it, you can at least go share this with doctors. And that Amarin EPA product, FACP, has skyrocketed in sales because doctors believe this. Hey, additional data with the FACP. So the company and so you would add FACP to your favorite statin of choice, and then you're
Which
taking
statin do they make?
No, they don't make a statin. They make EPA. So they want you to add FACP to whatever statin you're.
I thought they also made a statin.
No, they don't. Oh. So you can pick your favorite statin, but now you have like a ezetimibe. You might add to a statin. You can now add FACP if you're evidence-based, you're going to follow the trial.
I'm going to wait till November.
Oh, I would too.
Yeah.
And not that I wouldn't be using EPA, DHA for perhaps other reasons, cognitive or whatever, just cell membrane health. But the other company that makes a statin called rosuvastatin, and that would be called AstraZeneca, is the one that owns this. So what they were clearly hoping that had Amarin got an indication to add it to a statin, they would have released a combo product called rosuvastatin, their EPA product or so. And you would have in one pill your statin plus your proper dose of omega-3s, and they would be free omega-3 fatty acids, which would zoom right into your body. I got a feeling and by the way, they are completing their own big outcome trial with rosuvastatin plus the non-esterified free fatty acids. So they'll wait for their trial too. But if this first trial works, why would theirs not even work better? Because it's combining it with the most potent statin. So all these people who have been bad-mouthing omega-3s with no trial data may have to change at least some of that talk come November at the American Heart Association. It's the biggest trial going to come out of AHA this year, at least in the lipid world, so pay strict attention. Will a month or so before. Normally they don't leak out that data because that somehow prohibits publication in prestigious journals, but some companies do because it just means so much financially to them that it'll get published somewhere. So I don't know. One way or another, we're going to have it in November.
Now, we've danced around this idea of red blood cells for a while. They keep coming up. So let me redirect us to another topic, which is you've already said red blood cells per molecule carry many, many more molecules of cholesterol than any lipoprotein does.
Right.
Why don't red blood cells cause atherosclerosis?
Because they're not invading your arterial wall and not subject to oxidative forces where a macrophage is going to ingest the red blood cell.
Do they not invade the artery wall because they can't penetrate the subendothelial space?
Yeah, they're gigantic. Now, listen, can a red blood cell get into plaque? Yeah, through the vasovasorum. So could there be some in there that are...
So the vasovasorum, meaning from the other side.
Yeah, well, remember, artery walls get arterial supply. They need oxygen. And even plaque has little capillaries and stuff. So are there red blood cells in expanding plaques? Sure. So not to say they're not a tiny little part, but that's not your primary delivery of oxidized phospholipids and sterols into the artery wall. Red blood cells are not entering the artery wall and being oxidized, like the ApoB particles are.
Yeah, so this kind of gets back to sort of the causality issue of low-density lipoproteins.
Although also red blood cells carry way more cholesterol molecules, you have bazillions more ApoB particles than you have red blood cells. Also, so yes, quantity-wise, they're carrying a lot of cholesterol, but not red blood cell number-wise versus LDL number-wise.
So the size of the LDL particle, obviously, is what enables it to easily get into and out of.
Number and size, but as you know, I've taught you this a long time ago, any VLDL or under 70 nanometers can work its way into an endothelial gap or an arterial wall through a scavenger receptor, whatever. So not to say remnants aren't atherogenic. They can't get in because remnants are way bigger than an LDL. No, they could. But there's just more of the LDL particles. So if this is a diffusion gradient forcing them in, there's a hell of a lot more LDL troublemakers than there are remnant troublemakers. Not to say some big tough guy isn't a big troublemaker by himself. He is, but which would you rather have, 1,000 guys coming at you or one guy? I'd take my odds that I could beat one guy rather than 1,000 guys,
Yeah. I figure it's hard to believe we've been going at this for almost 7 hours, and I feel like there's at least another hour or two we could go, but at the same time, I want to be sensitive to the listener. Normally I say I want to be sensitive to the guest, but I know you and I could keep talking about this for another 6 hours. Right. And we're going to go have dinner with Jamie Underberg tonight and just continue this discussion. I wish I had a microphone for dinner because I have a feeling that's going to be equally interesting. One of the things you talked about at the very outset was a really close friend of yours who got you into hockey. Took you to your first game at Madison Square Garden, got you hooked. You were both obsessed with being firefighters. He went on to become a firefighter. You went on to become a lipid educator.
Yeah, so let me just pick up that if you look back in life, who are some of your absolute best friends, your high school buddies, who you really spend all that the type of enjoyment you can have at that age that you never can have at another age, and you can do things, and especially when we grew up and we were in high school, 59 to 63, you could get away with a lot of junk in those days, whereas today we were expelled real quickly or so with some of the shenanigans we do. So, and look, I joked with Peter when I was telling this story. I think he really wanted to be my best friend because my father was a fire chief in the town of Paterson, New Jersey, where we went to high school. But even if that was true, I love firefighting. What do I care if he loved firefighters? We used to get on our bikes and ride to fires all the time together and watch fires and stuff or drive to firehouses. So we were really tight for a million reasons, and we just got along personality-wise. But he's the guy who invited me to my first ice hockey guy, which was certainly impacted my life and my son's life in mega ways. My son's a very successful guy, and he would tell you he would never be half as successful had he not played ice hockey and developed the teamwork and the camaraderie. Speaking of a fire engine.
It is perfect timing to have a fire truck going down.
But I'm going to just keep talking about this fire truck because it's not high lipid science. So my buddy there, he went and became, and I knew him all my life, and I went and became a doctor, but right in that same city or in a suburb of that city, so it's not like he didn't know where to find me. But at a certain point early in our life, I knew my buddy, hey, you're a doctor now, I got this cholesterol problem. They tell me my cholesterol's up in the blood. And, I was probably at the point where we were really taking cholesterol serious and we could do something about it. I said, you got to come and see me. Come on. And I was probably even the days before I was just solely dwelling on lipoproteinology and lipidology, but I was very aggressive with it, understanding cholesterol, knew it, and look who I evolved into. So it would have probably been a smart choice for him to come up. It's not like I was going to charge him and he couldn't afford me. He's a fireman in a big city. He had as good a health insurance as you could ever get. So it's not like it would have cost him 10 cents. A lot of firemen did pick me as their doctor because of who my father was, and they knew he was a guy who likes fire engines. You used to see him as a kid in the firehouse. So I want him to be my doctor. He knows what I do. Earl just is one of these people who thought the farther I stay away from doctors, the better. I don't want to hear what they got to tell me. I don't want to be on a medicine. I don't want to do anything. And Earl got buried in his mid-50s with a sudden acute myocardial infarction sitting at home watching TV. He could have been climbing a ladder at a fire. By the way, if he did die at a fire, his name would be on the memorial monument up in that city. But he died at home the next day. So not that I want his name on I really wish it was, because he's just such a death. But there is such a needless death. And I think across America all the time, there are many young age needless deaths occurring with a very preventable, treatable disease that's being ignored totally because they don't go and get it checked, or they're getting bad advice based on the wrong metrics. So my buddy there is the perfect example. But what a tragedy, because had he made the right, I like this guy, he's my best buddy, he's not going to screw me or do anything wrong, let me go to him. He didn't. Every time I saw him, I'd remind him, Earl, come up, please come up. Yeah, Tom. Never did.
Well, in many ways, that's a reasonable and sad way to kind of end this discussion, but it also brings it kind of back to why do we get so animated about this topic? Well, I think, as you said, we think of this as, one of the big three diseases that kills most people in the civilized world once they get out of childhood. And by civilized, I really mean developed. People know what I mean. But of the big three diseases, this is the one where I think we know the most about it. And therefore, by extension, it's probably the one that's most delayable. Never want to say preventable, right? We use that term, but in reality,
Correct.
What does that mean? It means delay. And so if Earl died in his early 50s, maybe with the right care, he could have died in his late 60s. Again, if a guy's dying in his 50s of heart disease, he's probably got really bad disease.
He needed therapy at a real young age, but he could have hung around for a while, and he's got grandkids and children and stuff who really wish he was still here at the fire department. He was a beloved fireman. They all wish he was still on act. He'd be retired by now, but he's just one of these people like God sends to earth that do nothing but good while they've been here through their whole life. He had saints as parents, and it was so tragic that he left us at such a young age and got driven to the graveyard on the back of a fire truck with a flag on it, Vietnam veteran. And but just so sad. And listen, I want to wrap this up myself. And I used to wrap up a lot of lectures I did with this. And, because my lectures were all high science, no matter what group I was speaking to. So I always wanted to say, you ought to come out of this lecture that I give, and maybe with all the things, if you really sat through everything we talk about today, to say, guy's an idiot, or he's on to something, and I thought I understood this topic, but maybe I need to understand it at a higher level. So everything is study. I didn't learn this stuff overnight. My life has been spent in this field.
Yeah, well, I'll say a couple things. I'll say a couple things on that. So first of all, I obviously want to thank you for the influence you've had in me, and I don't know, I just feel really lucky. I think that I got plugged into you and Alan and Ron just out of the gate to have met the three of you so early in my interest in this topic saved me so much time, because it's one thing, no one can substitute the amount of time you have to spend obsessing over this topic and learning it. But boy, when you can have that curriculum curated for you, you shortcut it by, 3x, I'm sure.
It's a gift, and I'll just extrapolate on that because Peter named three guys he can get in contact with pretty quickly and get answers. As I evolved in this world, I had 20 guys around the world who liked me a lot because of the way I explain things, what I could illustrate for them. So I've been blessed by, I've mentioned a few today. It would take me another 15 minutes to list them all, but so many of I call them the lipid gods out there, the people who have spent their real life in research laboratories and clinical trials and taking care of people in the worst lipid clinics, and they still are available to me. But that's who I use. I don't go on the internet and read something or make stuff up. I try and ask experts about a lot of things. Not that you'll ever get consensus on anything, but those of us who do have those avenues open, which many do not, but they're not afraid to still pontificate about something. It's kind of sad.
Yeah, and I can't remember if I made this will be my final point on the topic, and I can't remember if I made this point on a previous podcast or not, and if I did, to the listener, I apologize. I started out in mathematics and engineering and really loved those things. I excelled at those things, took great pleasure in being a first principles thinker. So one silly story, at the end of my freshman course in sort of dynamics, kinematics, like sort of Newtonian physics, you take a final exam and you are allowed one piece of 8x10 paper that you can write on both sides as much as you want, whatever formula you want. Because, there were just so many equations you would have to know to go in and ace one of these exams. Now, I was cocky beyond words. And I was really lucky as a freshman. I used to study in this dark part of the stacks where I met a real upperclassman guy. His first name was JP. I don't remember his last name, but I'll never forget him. He was the only guy I'd ever met who decided to take 2 engineering disciplines and finish them both in 4 years. So he did electrical and mechanical engineering in 4 years. The price he had to pay for that was he studied every minute of every day. But he also told me he didn't have the opportunity to learn everything because you couldn't. If you wanted to take all of electrical and mechanical in 4 years, you had to accept that there were certain things you couldn't know. So all of his study focused around first principles inference. And he taught me this early. And so he saw me studying for one of my exams, and I was writing out something called the Coriolis equation, which basically explains centrifugal forces under the condition of a changing radius. And he said, "You don't need to memorize that formula. You know how to derive it." And I was like, "What? What are you talking about?" He goes, "Write out the formula for angular position and take the derivatives of it. You know calculus." So I did. And sure enough, I was getting the same formulas. To make a long story short, I've become totally inundated by JP's reasoning. I go into my final exam and, you have to turn in your cheat sheet at the end of the exam. And this is again, I'm not saying this to brag about how cocky I was, so I just have to stay. But the point is, I wanted to show the professor how well I understood this material. The only thing I wrote on my cheat sheet in big block letters was the sum of the forces equals mass times acceleration. Because I was like, if you know the simplest of Newton's laws, you can actually derive everything at this level of physics. We're not talking quantum physics at this point. And I remember JP said, "Look, on every question, write it out. Show them how you can go from one to the other. Because if you make a mistake, at least they'll see what you're doing." I ended up acing the exam, and that lesson sort of stuck with me, and it served me very well through the rest of engineering and mathematics, including when it got really, really complicated. That in the end, if you truly wanted to understand something at first principles, you could derive so much. Okay. Fast forward a few years, now I find myself in medical school. Now, I was not a pre-med. So I'm in medical school, not as a pre-med, but as a former engineer math guy who barely squeaked into medical school, meaning I took the MCAT having not even taken a course in biology yet. I had to take my bio class after. And I'll tell you, that first semester of med school was brutal for me because I learned the hard way that you couldn't first principles your way through biology. At least not at the level, at least not until you gained a certain amount of familiarity with the basics. I couldn't walk into my histology class, look under the microscope, and on first principles know the difference between the Golgi apparatus and the endoplasmic reticulum. You just have to know that stuff. I couldn't go into anatomy and physiology and pathology and just first principles my way into stuff. Eventually you can, but you have to have an enormous basis. So I sort of forgot all about that pain. You get through medical school, and by the end it became, it was very enjoyable, but it wasn't until I got to the lab to NIH where my mentor told me a great story. Now, he's probably one of the most brilliant scientists I've ever known, and did a PhD in biophysics specifically for the purpose of wanting to make sure he was never, in his words, intimidated by a differential equation, despite the fact that he was an immunologist. And he told me a story, which I need to get the details of it because I don't remember the physicist he's referring to, but he told me the story of a physicist turned biologist who lamented once that the moment he switched from physics to biology, he could never again take a bath in peace. Because as a physicist, he could sit in the bath and wax philosophically and think about lots of things on first principles and be very theoretical in his understanding. But as a biologist sitting in the bath, he always realized there was one more fact he needed to remember before he could draw a conclusion. And so he had to keep getting out of the bathtub to go and look up a certain fact. And that obviously disrupted the beauty of taking the bath. And I think the long rambling story is a way of saying the following. I find this subject matter to be as complicated as any I have ever tried to learn. And unfortunately, every time I have a brilliant idea just based on first principles, I still have to go and check if it's grounded in the foundation of science. And sometimes it is, but many times it is not. I come up with an idea and I'm like, this has got to be right. Oh, it's physiologically impossible. And there's even an experiment that one day suggested this, or it's, I made an assumption, but I can clearly see that it was not the case. So I understand your frustration and I share it. And I think, unfortunately, that is the nature of biology.
Yep. And I remain an eternal optimist. I continue to teach lipids. I think clinicians know a bit more than we knew in 1995 when I started this journey teaching. And so that's all. If I have to do it one at a time with people, I try and teach them what I know. And hopefully you'll do your continuing education. And that's one of the nice things about medicine. But you got to do it. None of this comes easy. It does. And there's a lot of complexities in medicine nowadays. We're talking because it's a highly pathologic state and leading cause of trouble, but there's so many other things to, take to the much higher level nowadays,
Well, Tom, people, I think, will certainly by the end of this know where to find you, but on Twitter, which is where you spend the most of your time, you're at Dr. Lipid, correct?
That's me. So, yep.
And we're generally in our brief existence as a podcast already known for probably producing the most thorough show notes ever. I'm suspecting that this episode, which will probably break into several parts, will have the most robust show notes that we've done. And I love, by the way, that there's another fire truck going down. It's perfect. It's totally appropriate. Yeah. But also, if there's anything else that you think of, let's just talk about it where, offline, even we can just get to it, where there are other resources you think people ought to go to. Again, we'll steer people away from your 2000 to 2004 writings. But I think there's going to be a number of people who listen to this who say, you know what, look, I might have missed half of what those guys talked about, but there's something there I want to make sure I understand better, because in the end, every doctor wants to do the best by their patient. I mean, that's just, I believe that wholeheartedly. Yeah. I get very frustrated when I hear people say that doctors are in the pockets of pharma companies and they're just in it for the money. I mean, that's, sure, there's going to be some of those people, but I haven't met those guys. The doctors that I meet, the men and women I meet who take care of patients, they all want to do the best by their patients. And I think that those are people who are going to want to invest a little bit of their time in understanding this problem a little better. No, I can't say it any more than that. And as you listen to 7.5 hours, even hopefully piecemeal marketer, clearly depending on the list, there's going to be things over your head. Even if you are a super skilled lipidologist, there's going to be some things you said, what? Let me read about that or so. But it's just trying to open your mind and understand there's a lot going on here. The field is infinitely more complex than you ever imagined. It took me a long, and as Mike Davison calls me, Tom's a self-taught lipidologist. I mean, I didn't spend 2 years down at the National Heart, Lung, and Blood Institute, even though the director of that NHLBI, Alan Romali, and I just authored a 70-page chapter in the Teats book of molecular and clinical biochemistry, all about lipids and apoproteins. So I think to be an invited author with a guy at that magnitude. And you sent me a preprint of that. That's a remarkable treatise. I mean, it's an expensive book. I don't expect you, if you're not a lipidologist or somebody seriously into this clinician, that's what you go out and buy. But there is a lot of information out there.
Just be careful on that's ridiculous, or my cockamamie theory based in no clinical trials or science is what I believe. Fine, believe it. This is America here. You're free to, I can't lock you up. I'm not the lipid police.
Yeah, well, on that note, Tom, thank you very much. It's hard to believe this has been 7 hours of discussion today, but it's been worth every minute to me. And I've learned a lot in this discussion. So to me, that's always for selfish reasons, that's sort of my primary objective is to learn as much as I can. And then along the way, if others can learn too, that's a bonus.
And without a year of my intermittent fasting and program from Peter Attia, I could have never had the energy to sit here all day long, even to use my tongue or brain. So thank you again, Peter.
Yeah, my pleasure, Tom. Thanks. You can find all of this information and more at peterattiamd.com/podcast. There you'll find the show notes, readings, and links related to this episode. You can also find my blog and the Nerd Safari at peterattiamd.com. What's a Nerd Safari, you ask? Just click on the link at the top of the site to learn more. Maybe the simplest thing to do is to sign up for my subjectively non-lame once-a-week email where I'll update you on what I've been up to, the most interesting papers I've read, and all things related to longevity, science, performance, sleep, etc. On social, you can find me on Twitter, Instagram, and Facebook, all with the ID Peter Attia, MD, but usually Twitter is the best way to reach me to share your questions and comments. Now for the obligatory disclaimer. This podcast is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. And note, no doctor-patient relationship is formed. The use of this information and the materials linked to the podcast is at the user's own risk. The content of this podcast is not intended to be a substitute for professional medical advice, diagnoses, or treatment. Users should not disregard or delay in obtaining medical advice for any medical condition they have and should seek the assistance of their healthcare professionals for any such conditions. Lastly, and perhaps most importantly, I take conflicts of interest very seriously. For all of my disclosures, the companies I invest in and/or advise, please visit peterattiamd.com/about.